First of all, I just want to say thank you all for coming. It's really great to see so many of you here tonight. I'm only LA for a couple of days, and I don't come here nearly often enough, so it's marvellous to see such a great turnout. Hands up everyone who was totally snowed by that last talk, for who it was totally over your head. Come on, be honest, be honest. All right, now that's good, right? It's really important to me that people understand that we're doing stuff that you don't understand, that we're beyond the point of just telling you what our plans are. Okay, it's really important. Jacques didn't make much of a show of it, but the fact is he's been working with us for 5 or 6 years, and that's how long it takes to get really impressive results in any really difficult pioneering research field. And biomedical research like this is no different. The fact is, we've got now to a point in the research program that Jacques has been working in that has allowed us to publish results that are really pioneering. One slide that Jacques put up showed the protection of cells from the major toxin, this thing 7-ketoglycerol, that drives cardiovascular disease. Now, you all know that cardiovascular disease is the #1 killer in the Western world. But let me be perfectly clear what this means. This means that we are a very significant step towards eliminating, not just slowing down, but eliminating the #1 killer in the Western world. Huzzah! Now, most of the projects that we work on have not been going nearly as long as this. One or two of them have, and still moving forward. Some of them are even harder than what Jacques is working on. I should briefly mention the other work that's going on at the research centre that we have. Our headquarters in Mountain View. The work that Jacques briefly alluded to towards the end of his remarks on macular degeneration is going on there. And the way that we're addressing that problem is very similar to what Jacques described. We're looking for enzymes, whether human or more likely non-human enzymes, which can break down the substance that accumulates, again, within the lysosomes of the relevant cells, in this case retinal pigmented epithelial cells, and causes pathology, in this case macular degeneration. We're not quite so far along as in that project as we are in Jacques' project. We haven't shown increases in cell viability, but as Jacques briefly explained, we're getting there. Another project that's going on in the lab is headed by another of our researchers here, Mathieu. Oki, are you here? Oki? There he is in the back there, so if you want to know more about this project, you can ask him. We're going to be here for a little while after I shut up. That project is all about addressing the problem of accumulation of mutations in the mitochondria, this incredibly important part of the cell that does basically the chemistry of breathing. We have this crazy idea, which actually was not originally our idea. It was first proposed in the mid-1980s, of making the mitochondrial DNA unnecessary by copying it with suitable modifications into our normal chromosomal DNA. And great progress being made there, which Oki will be happy to talk to you about. And there's also a project going on looking at eliminating cancer. I'm sure all of you have heard of telomerase. Hand up anyone who hasn't heard of telomerase. All right, not many, all right. So this crazy enzyme that is responsible for allowing cancer cells to grow indefinitely, and therefore for cancers to get big enough to kill us. And 85% or so of cancers in human beings express very high levels of this enzyme, which allows them to do that. Without an ability to do what telomerase does, namely to extend the ends of chromosomes, cancers just would never get more than a millimetre or two wide, and they just wouldn't kill it. So that would be great. But 15% of cancers do what telomerase does without telomerase. They do it in a different way. And the third project that we are pursuing in our lab in Mountain View is to find out what's going on there. This has been a really neglected area within cancer biology. There's only a couple of labs in the whole world really working on it, and they are Well, they're great labs, but they're definitely not covering all the bases, and we're doing stuff that is not being done anywhere else and which could, in due course, make a really big difference to the elimination of cancer, which is, of course, an enormous part of the elimination of the ill health of old age. So that's what we're doing in the research centre. But the reason that we put this slide up here is to emphasise and to remind you all that we don't just have a research centre. I don't know the exact numbers as they currently stand, but I think it's roughly that out of our research budget, 1/3 or so goes on the research centre, and 2/3 goes on extramural research. That means we fund laboratories around the world, mostly in the US, that are at universities doing particular work. So Jacques' work is an example of this. Jacques and a PhD student working in the same lab, Jason Gasper, are working on this lysosomal enhancement problem, LysoSENS. But as you can see on the right-hand column there, we have a bunch more. I think there are 12 things there that we're listing. I'm going to have to get closer when I do these in a sensible order, and I can see the screen. Here we go. All right. So I'm going to do this in a vaguely scientifically sensible order rather than the order that they're written down. So, as many of you know, SENS consists of a dissection of the problem of aging into 7 subproblems. 7 types of damage that accumulate throughout life in the body and which eventually cause the pathologies, the illnesses, the disabilities and degeneration of aging and eventually kill us. And all of these 7 things are in one or another way represented in the work that we are funding, both intramurally and extramurally. I just mentioned one of them, the accumulation of mitochondrial mutations. The other one that happens as molecular changes inside the cell is the one that Jacques spoke about, the accumulation of junk, basically, of garbage inside the cell, typically in this place called LysoSENS. And Rice University, which is where Jacques works, is mentioned in the middle about this, as you can see. There's also molecular damage that accumulates outside the cell. And 2 of the problems that we are working on in SENS Foundation, 2 of the 7 strands of SENS, are of that character. One of them is being pursued as a collaboration between a group at the University of Texas in Houston, towards the bottom about this, and also at Harvard University. I think it's probably discussed. Yeah, there it is, Harvard Brigham and Women's Hospital, which is an affiliate of Harvard University. Here we're looking at trying to eliminate a type of protein that accumulates in the spaces between cells in many tissues in the elderly, especially in the heart. And it has been established, especially due to the wonderful work of a guy named Steve Coles, who is here tonight, and who has done the relevant autopsies, that most people who die at an extremely old age, 110 or more, tend to have this as their major cause of death, and thing that mainly kills them. Their hearts basically stop beating because they've got this stuff accumulating between the cardiac cells, between the muscle cells that make the heart up, and it just gets in the way. And we'd like to get rid of that stuff, and the collaboration between the University of Texas and Harvard is doing that. The other extracellular problem, the problem of material between cells as opposed to inside cells, consists of changes in the physical properties, the actual mechanical properties of the lattice of proteins that holds us together. There's a bunch of proteins that are linked together in a very regular pattern, a lattice. It's called the extracellular matrix. And it's responsible for holding our organs in the shape that they need to have and so on, and having the physical properties they have. In particular, it's responsible for elasticity, which is a critical component of how certain of our organs behave. You can think about, for example, presbyopia, the fact that in old age you can't see things close up so well. That's because the lens of the eye gets stiffer. It's not so elastic as it used to be. The same thing, in a much more life-threatening way, happens in the walls of our major arteries. We get stiffening of those arteries, and that is what causes hypertension. That's the main reason why blood pressure is a problem in the elderly. And of course you know all the things that causes it. Exacerbates diabetes, for example, loads of problems. We really need to fix that. And we have a very major project going on right now, which is a collaboration between two more of the things on the list on the right there, Cambridge University and Yale University, looking at the identification of molecules or other therapies that can restore that elasticity, that can break the chemical bonds that are accumulating in the extracellular matrix throughout life and that cause the extracellular matrix to become stiffer. On top of that, we have, so I've listed now 4 molecular changes that happen in the body, 2 inside the cell, 2 outside the cell, that are 4 of the 7 strands of cells. The other 3 strands are cellular. They're not intracellular or extracellular, they're at the level of cells. And what I mean by that is that they're at the level of the number of cells we have, not the function. So, of course, cancer is a case of having too many cells. Cells dividing when they're not supposed to. And I already mentioned one case of, one aspect of how that can happen when cells get into this state where they can extend the end of their chromosome in a way that's poorly understood right now because it does not involve this well-understood enzyme telomerase, and we're working on that. But in addition to that, there is a second way in which we can have too many cells, which is if cells don't die when they should. Most people don't think about that. They think, why should cells ever want to die? Why would it be good for cells to die? But it turns out there are actually really important aspects of how the body works that depend on cells dying. One of them is the way the immune system works. When the immune system encounters a new infection, there's an enormous amount of cell proliferation, and that's what we need to get rid of the infection. But then after the infection has been gotten rid of, we've got to get rid of most of those cells again, so as to make room for cells to divide again for a new infection. And that doesn't work so well in old age. So at the University of Arizona, which is listed here in the middle, we are looking at exactly that problem. And actually that's a project that's been going for a couple of years now, and it's pretty close to publishing some interesting results. So again, I mentioned that Jacques and his group have published a very nice paper just a few months ago summarizing the status of that work. Many of the things that we have been working on, because they've only been going a couple of years, they haven't really got to the point where we can publish anything yet. But some of them are getting very close. So watch this space. In the coming year, we're going to see a lot of publications on some of these projects that we're talking about here. The third thing that we need to worry about with regard to cell number is having too few cells. And that happens when cells die and they're not automatically replaced by the division of other cells. That's the reason, right? Cells die and they're not replaced because you have too few cells. That turns out to be important in a large number of aspects of aging. Parkinson's disease is one example. We're not working on that, other people are working on that, and we're always very careful to get the best bang for our buck, to be working on things that other people are neglecting. But we do have a number of things going on in that area. In particular, at the Wake Forest Institute for Regenerative Medicine, one of the most prestigious institutes in the world working on regenerative medicine, we have 2 programs going. One of them on the thymus, which is another thing that is part of the decline of the immune system. In this case, it's where the thymus just loses cells and ends up in a shrunken state where it can't process new immune cells properly. And we have a second project looking at the gut. The gut is an area where not much goes wrong during normal aging, but we anticipate that plenty will go wrong as a side effect of what we want to do against cancer. So we're very interested in ensuring that we have ways to address those side effects. I've gone through most of the things on this list now. There are really only 3 that I haven't mentioned. And they are what we're doing at Einstein, the first thing on the list, what we're doing in Denver, which is number 8 or so, and what we're doing at the University of Arkansas, number 7. And those things are actually things I want to spend my last couple of minutes on, because they're not what you would obviously expect us to be funding. We have this project plan. It's called SENS. It's this dissection of aging into these 7 components and implementation of ways to address all of these components. But why this particular plan? we are absolutely cognizant all the time that we need to know, and we need to justify, and we need to be sure that we're following the right plan. And there are 3 reasons why we might not be. And we need to be looking at all of those reasons all the time. One of those reasons is, it might not actually be a comprehensive plan. There might be other things going wrong in aging that none of these 7 proposed therapies will actually address. And that would be a bit of a shame, because we could grind on and develop all these terribly ambitious therapies and get them all working, and people carry on dying on schedule. That would be pretty sad, really, because there was something we missed. So we don't want that to happen. Second possibility is that SENS is too complicated. That there's actually a simpler way, by which I mean a way that one might be able to implement more quickly, that will achieve the same end, that would actually postpone aging by the same sort of amount. If we could do that, wonderful. I've got no ego in this. I just want to save lives. I just want to alleviate suffering. And the same applies to the whole of this community, I'm sure. Certainly everyone in SENS Foundation. So we always want to be looking out for possibilities that we might be able to do better. The third thing that we always have to focus on is rollout. It's all very well developing the science. We want to make sure that the science is developed as quickly as possible and is disseminated as quickly as possible, so that it saves as many lives as possible. So all of those 3 things, I've got into the habit of calling that family of things that are not really SENS, I call them meta-SENS, but I'm not sure anyone really approves of that name. However, those 3 things I just mentioned, what we're doing at Einstein, at Arkansas, and at Denver, they each fit into one of those 3 parts of meta-SENS. In Einstein, we're looking at the phenomenon that is most often raised as a possible 8th thing, something that might be accumulating during aging to a detrimental degree. We think it probably doesn't accumulate to a detrimental degree, but we're checking. And this particular thing is the accumulation of what are called epimutations, changes to not the sequence of our DNA, but rather the way in which the DNA is turned into protein. This is something that people have been very interested in for a long time. In order to study it properly, they had to develop an incredibly sensitive technique that was 1,000 times more sensitive than what existed before. They've done that, and so now we can ask the key question we want to ask. And this is a really major project of ours. In Arkansas, we're looking at a phenomenon in worms, in tiny little nematode worms, that appears to be a hint that it might be possible to do something that's simpler than SENS. We think that probably this is a false alarm, so to speak. It's not actually true. But we've worked out, I realized that there was a way to test whether this was actually a false alarm, and that's what we're doing in Arkansas. And in Denver, we're looking at the social and political and particularly economic consequences, especially as regards to demographic consequences, of actually developing these therapies and having them roll out. The reason we're doing that is largely to dispel most of the fears that politicians, policymakers, and of course funders have with regard to the consequences of seriously defeating aging. This is something that hasn't been done properly in the past. The group that we're funding in the University of Denver are an extremely prestigious and well-respected group that people will listen to. So we are very keen to make sure that they produce, obviously, a completely unbiased, but nevertheless authoritative analysis of what's really going on. So that's what I would like to say tonight, to emphasize the projects that we're doing. As you can see, for those of you who have been following SENS Foundation's work for the past several years, we have diversified a lot in recent years. We've been able to grow the portfolio of our research program enormously. I'm absolutely overjoyed that we've been able to do that. At this point, to be honest, as we grow further, what's going to be going on is going to be of a different style. So far, in growing from, let's say, a budget of $1 million a year to $4 million, which is what we are this year, we've been expanding the number of projects, making sure that we cover all the bases. Going forward, we sure as hell need more money. We could spend 10 times more money than we have, no question. But most of where that money will go will be in expanding these projects rather than in starting new projects. All of the things I've been describing to you tonight are funded, but at what we would call entry level. You know, we could certainly be doing far more in most of these areas, getting results far faster. People often ask me about time frames. People say, how soon are we going to have these therapies? I've said since about 8 or 9 years ago that it was probably going to be about 25 years before we got these therapies to work. And I always emphasize, of course, that that prediction is extraordinarily speculative, but that's just a 50-50 guess. I think we've got a 50% chance of getting these therapies online within 25 years, but I completely accept that there's at least a 10% chance of not getting there for more than 100 years. No problem, you know, a 50% chance is quite enough to be worth fighting for. I've also said, since about 8 or 9 years ago, that it would take about 10 years to get to the equivalent degree of success in mice. Okay? Now, you may have noticed that we're nearly up to that 10-year point. Sounds bad, doesn't it? Sounds like I was very over-optimistic. But no. The thing is that right from the beginning, I always emphasized that we would only be able to achieve that rate of progress and that sort of time frame with sufficient funding. And we haven't had, obviously, sufficient funding. I was always talking in terms of like $100 million a year, and we haven't had that. I feel that the amount of progress we have made in those past 8 or 9 years is about 1/3 of what I was hoping for. And that's pretty much exactly what I would have guessed with the amount of funding that's actually been forthcoming. So I'm actually pretty happy that I was not either over-optimistic or over-pessimistic. This is looking pretty good now. And all we need is to ramp up the funding and we'll really be able to get some action. We've made 3 years of progress in the last 9 years, and that's not to be sneezed at. But let's make 3 years more of progress in the next 3 years. Thank you.